Hello, everyone. In this video, I will try to set you up for a paper and pencil activity to help you appreciate what is meant by reverse engineering the brain. Doctors, psychiatrists, engineers, and neuroscientists are all actively participating in brain research. All of these perspectives are beneficial and further being explored to help improve our modern understanding of the brain. In fact, the National Neuroscience Curriculum Initiative is an initiative that promotes a collaboration between educators and neuroscientists. The overarching aim of the National Neuroscience Curriculum Initiative is to create, pilot, and disseminate a comprehensive set of shared resources that will help train psychiatrists and other mental health providers to integrate a modern neuroscience perspective into every facet of their clinical work. So therefore, we will try and look at this activity through a circuit, engineering, neuroscience perspective, while also understanding that these mechanisms may have further implications in medical and psychiatric treatment. In this lesson, we will discuss Pavlovian conditioning and watch a 1-minute video of a Pavlovian fear experiment occurring in rats. It will be good if you take notes about the details related to this 1-minute video that I will explain first. The details related to the process of rodent fear conditioning will help you reverse engineer the fear circuit yourself at the very end of this exercise. So let me explain what you will see in the video, and then you can load the video file and watch it at your own pace. So the video was developed by Professor Gregory J. Quirk of the University of Puerto Rico, San Juan. The video shows 3 phases in the Pavlovian protocol. First, habituation, which is where the rodents get used to a high-pitched sound. Initially, it makes them anxious, but then they get habituated to the sound, meaning they get used to it. And the video does not have a sound, and the onset of the high-pitched tone is indicated by a light coming on. So please remember that. The video file does not have sound, but you can tell when the high-pitched tone is present when you see the light coming on. So the next phase, the conditioning phase, is when the rodents receive a foot shock that co-terminates with the tone. So that is, the shock is administered when the tone is on, and the tone and shock are paired. Now, during the final phase, which occurs 24 hours later, there is the extinction phase, which is when the rats only experience the tone being played and do not receive a shock. And this phase is known to diminish fear for normal rodents, and deficiencies in reduction of fear in this extinction phase are thought to underlie human anxiety disorders such as PTSD. Another point to note in the rodent experiment is that different levels of freezing indicate different levels of fear in the rodents. That is, hungry and caged rodents express strong fear by complete immobility, and lesser levels of fear by cautious movement, and so on. So different levels of fear can be measured in a principled manner in such an experiment and done automatically by software that can record durations of rodent immobility. So the clip has 4 videos of 4 rodents in different cages, all fused onto one screen. So we have an N, or a subject number, of 4 in this experiment. Now, that was a lot of information, but try to remember that as you watch the video, you can pause and go back, and you can also review the information I just described. So pause this video now, and then watch the behavior video of these rats, that 1-minute video clip, then come back and resume this video. So I hope you liked the video. And if there are parts you were not clear about, please rewind it and try to watch it again for specifics. Your paper and pencil activity will involve figuring out how fear was learned by the rodent brain in that video. That is, what part of the circuit in the rodent brain changed that caused the tone to elicit a fearful response, and how did that happen? So, not sure if you've thought about this, but fear learning happens unconsciously in animals. And in the rodents you saw, it is similar to how fearful events may have happened in your lifetime or in any individual's lifetime, and how it was remembered and stored. So that is, we learn fear without being consciously aware that we have learned it. So before we move on to the paper and pencil activity, let's step back a bit and build up to it, but step by step, by starting with how all living creatures may be hardwired to learn two key biological traits. So we will then provide you with some specifics related to what is known presently about the mammalian fear circuit, what is learning, and about what exactly you were supposed to sketch. So let's move on to those slides now, and let's start with this question about what are two key traits seen in all animal species, ranging from insects to crabs, rodents, and even humans. So I want you to think a little bit about this to yourself, and then I will go over it. So the two key traits seen in all animal species are survival and reproduction. Now let's talk about the trait of survival that you and I also possess. So you may have not thought about this, but we are actually hardwired to survive and to reproduce. So how are we hardwired to survive? Well, one way to survive is to have innate fear. So some people or animals have fear of snakes. For instance, that makes us run when a snake is spotted. And that is a natural survival instinct. And this can happen without having ever seen a snake in your life. Now, how many such innate fears do you have? Now, it's something interesting to think about, but we will not talk about that since not too much is known presently about innate fears, or fears that we have naturally without having to learn. Another way we are hardwired to survive is that we have the ability to learn quickly about fearful events, and then we can adopt a fight or flight response to survive. So in psychiatry right now, trauma is a new and very researched topic that is a maladaptive mechanism of learning fear quickly and not being able to get rid of inappropriate fear responses. So going back to learning fear quickly, for instance, initially, if you see a hot stove, for the first time, and you don't have any indication of it being hot or warm, if you touch it, you will quickly learn that the stove is hot. And you only need to do it once to realize that you probably should not touch it again, and it will be recorded in your memory pretty strongly. And so that is an example of what we mean by being hardwired to learn fear quickly. So we have circuits within ourselves which are predisposed to learn quickly. So let's consider now the example of a rat learning to fear shock. So how does a rat learn to fear quickly? So what happens on day one? As you can see in this schematic, you have a rodent in a chamber, and it hears a high-pitched tone that goes with this blue tone release. And that The blue object or blue tone is a speaker, and the green you can see is a feeder. And the tone is given 5 times, spaced about a minute apart, so that the rat gets habituated to tone. And that was during that habituation phase when the rat got used to the tone. So this would be similar to you hearing music for the first time, let's say. You'll pay attention to it, and then after a while, you can recognize it, realize you've heard it before, and it's less stimulating or less new. So you become more normalized to the sound. So that's the habituation phase of the protocol. The rat is then taken to its home cage and brought back in a few hours for the conditioning phase. So in this phase, the tone is played as before, but there's a very brief foot shock provided at the end of each tone. The key point is that the tone and shock occur together. That is where the rat's fear circuit is learning that the tone is actually a bad, similar to you learning not to touch a hot stove. Note that the rat is learning to fear the tone without knowing that it is learning it. So this is unconsciously happening. Now, after this phase, it is taken back to its home cage and brought back on day 2 for the testing phase of the protocol. You need to know that a measure of fear in rats is that they freeze, as discussed earlier, if they cannot escape. And in this case, they're in a cage, they cannot escape, so freezing is their response, or is their fearful response. And that's not necessarily true in other animals or in humans. We don't necessarily freeze. So it's a little more complicated, where you have to measure skin conductance, heart rate, and other parameters of that fear response that demonstrate that the body has shifted into a response that is a fight or flight or fearful response. But with rodents, it's much easier, as they tend to just freeze when this occurs, especially when they're in a cage. So during the test trials, only tone is provided, so no shock. And as you must have seen in the video, during the test trial on day 2, the rats freeze as soon as the sound comes on. So note that during habituation on day 1, the sound was not fearful to the rat. So during day 1, when only the tone was played at the very beginning, the rats were not fearful of it. So they were not freezing. But after conditioning, the same tone on day 2 by itself generates fear in the rat. So if you see, there's a little green thing there in those schematics. And that green thing, as I mentioned earlier, is a feeder. And the rat was busy feeding. And you notice on day 2 during testing, the rat does not want to feed. So on day 1, the rat's here with this green thing. And on day 2, the rat does not want to feed because it is fearful. It's fearful of the shock following the tone, so the hunger response is suppressed. Now, if you provide many tones without shock, these are called extinction trials, and the rats will learn not to fear the tone. They feel that the tone no longer predicts shock, and so they continue feeding again. Does this mean that the fear memory has been erased? So that's what researchers thought initially. But then they realized the fear of memory is not erased, but it actually comes back via mechanisms such as spontaneous recovery, reinstatement, which we will not go into in this video. So we will not consider the extinction phase here, since that is a huge topic in itself. And we will address that in the next video, but it's something interesting to think about. We will only focus on habituation, conditioning, and testing of fear. So these top 3 conditions. So let us go to the next slide. What we'd like to do is reverse engineer the fear circuit in the rodent, which changed to predict the tone as being fearful. So what is meant by a circuit? This is similar to the electric circuit you see in a room. And in this case, we will consider the neurons and neural pathways that coded for fear. And that is what we mean by the fear circuit. We need a few biology basics related to this fear circuit before we can proceed. First, fear is linked to the firing of neurons in a region of the mammalian brain called the amygdala. Biological studies suggest that the amygdala nucleus is a key site for learning fear in rodents and also humans. So what does learning fear mean in this circuit? Can you guess? Probably you have read somewhere that learning has strengthened synapses. So these are connections between neurons in the brain. Now, how does learning strengthen synapses? What rules apply? So remember that the brain has to follow all these physical laws, and several laws you've previously learned in physics, chemistry, biology, and other undergraduate courses that are all applicable to the brain. So we won't go into too much detail about these laws, but just as a brief refresher, the Nernst equation, Ohm's law, chemical reaction equation, and electrical transmissions are all such laws that apply and are important. But we will focus on a law that you probably have not heard, but may have previously studied, called Hebb's law, which states that cells that fire together wire together. So your challenge is to first reverse engineer the fear circuit. That is, you are supposed to sketch the circuit and then determine how fear is learned. So let's give you some more basics before you sketch that circuit. What is tone and how is it perceived in the rodent brain? So if you consider your ear, you have a cochlea which has sensory neurons, and then the pressure signal due to sound is picked up by sensory neurons in the cochlea. So the sensory neurons then relay electrical signal via nerves to a region of the brain called the auditory thalamus, which is an important relay station for most sensory signals. So input that is received from the body. From the auditory thalamus, the signal is then relayed via firing of the thalamic neurons to the fear center, which we previously discussed, which is the amygdala. So the tone pathway is as follows. Tone is picked up via sensory neurons in the cochlea, then the firing of the sensory neurons relay that to the auditory thalamus, and the firing of the thalamic neurons transmit signal to the amygdala. So what about the shock pathway? Well, the shock signal reaches the amygdala starting at the paws or feet of the rodent, and the sensory neurons in the foot pick up the shock and fire, sending signal to the pain thalamus, which is a subdivision of the same thalamus that we had discussed with auditory, but this is a subdivision for pain signals. And the thalamic neuron, or the neuron in the thalamus, then fires and conveys the signal to the amygdala neuron. Now, the key point is that both the tone and shock signals converge onto the same amygdala neuron. So the tone and shock signals that went from the cochlea, sensory neurons, thalamus, and amygdala and the shock from the foot, sensory neurons, thalamus, and the amygdala, both of these are converging on the same neuron in the amygdala. So now we're going to go back to this tone and shock being paired and Hebb's law, which I had briefly touched on the last slide. And understanding Hebb's law first is realizing that when cells fire together, they wire together. So can you explain how this might be applied in the rat's case? So this slide here shows two pathways in the rodent. Don't worry about the details here, but focus on the two main pathways we described. The tone signal comes from the ear and goes to the thalamus and reaches the amygdala. The shock signal comes from the foot, goes to the thalamus and reaches the amygdala. And like we said earlier, the key point to remember was that Hebb's law states that cells that fire together wire together. And so we'll keep discussing Hebb's law, looking at this example, and come to a deeper understanding of what's going on. So now your first challenge is to sketch the fear circuit. So it should have two pathways, a tone pathway and a shock pathway, both converging onto the same amygdala neuron. Try to put one neuron in each pathway to represent each of the key regions. That is, one for the sensory neuron in the cochlea, one for the auditory thalamus, and one for the amygdala. Then there should be one for the foot neuron in the shock pathway and one for the pain thalamus, with that pathway converging onto the same amygdala neuron. See if you follow how 5 neurons should be enough for this simple fear circuit. We will represent each part of the circuit with one neuron for simplicity, although in real life there may be more neurons in the same signal. So in our circuit we sketch, we should have a total of 5 neurons. Now let us give you some information about the amygdala neurons and how they react to fear. So at the baseline resting state, when the rodent is not doing anything, the amygdala neurons fire at about 0.5 hertz, or at a rate of 2 spikes per second. In the fearful state, assume that the same amygdala neuron fires at 7 hertz, which is 7 spikes per second. The firing rates of the other neurons are provided in the slide. Make sure you understand them well. Another biological fact is that some synapses are plastic, meaning they are designed to learn and change, and some are not. And so they do not change or do not have any modification. So can you guess, in this case, what synapses in the 2 pathways you sketched may be plastic and change, and which ones are non-plastic and do not change, and can you explain why? So if you take it for granted that if the amygdala neuron is firing at 0.5 hertz and the animal is not fearful and can continue feeding, but if it fires at 7 hertz, the rodent will freeze and will not want to feed even though it is hungry. So you can take that for granted, and on a page, starting with a blank page, sketch 2 pathways, as mentioned earlier, the tone and shock pathways, which will have a total of 5 neurons in the various regions as discussed. And I'm only going to give you this information. So the rest is for you to think, discuss with others, and piece together. I will provide the solution sketch in a PDF separately, but it's best if you try to work through this and sketch it yourself for hour, couple hours, or a day to really just think about it before looking at the solution. I would also strongly encourage you to talk to others and even try to explain to others this concept to enhance your own learning before looking at the solution. And I will provide you with the answer in the next video. So congratulations on completing your first project in reverse engineering the brain. And I hope this will be the start of many more brain circuits you will reverse engineer. See you in the next video.